Okay, so this was a lecture for you. Um, no, that's okay. Okay, so this is a lecture on GLP-1 biotech and drug development. For those online, I'll probably post this lecture. For those online, I apologize for the microphone. It doesn't sound quite so good because my microphones have been having problems lately. Anyway, so who has heard about GLP-1 in the news? Anybody? Okay, so this is like the drug. This is, well, the GLP-1 receptor agonists are the drugs that they give to fat people to make them lose weight. The people who cannot lose weight, they give them this drug now, and it's become a very successful drug. Okay, so we'll talk a lot about the biotech involved in the development of this drug, and this lecture is based off four major sources. So there's two original sources where they did some real interesting biotechnology work by this person named John Eng, who was studying saliva proteins in the Gila monster, and he found agonists for the GLP-1 receptor in those creatures' saliva. And then I read a review on this, and then I also consulted one of the patents on the production of this drug. Okay, so to introduce the concepts that are important for this, to understand this lecture, so GLP stands for glucagon-like peptide. Okay, and I'm going to go through a couple definitions and explain some of the pertinent enzymes that you need to know about to understand the basic biology of what this thing is doing and what its context is. So GLP-1, that's a peptide hormone. So here is an actual sequence of GLP-1. So it's not a full protein, it's a peptide. It's a small peptide, it's about 30 amino acids long, and it starts with a histidine. Okay, this is a peptide produced by cleavage of a precursor protein called proglucagon. So if you look at the actual gene that encodes this peptide hormone, this is the actual construction of the gene, proglucagon. The actual gene probably has a different name, but this is what it's coding for. So it's coding for a large peptide or a large protein that has various parts of it that do different things that get chunkified up into smaller peptides. So often the case with like peptide hormones might be expressed from a larger gene, transcribed as a long message, and then translated into a protein. And then the protein goes through processing, which cleaves or chunkifies it into various chunks that do certain things. So this proglucagon peptide or protein gets cut up into various different parts, including GRPP, glucagon, major proglucagon fragment, which then gets truncated into GLP-1. And the GLP-1 actually has some unique modifications to it as well. So it comes from a larger protein that gets chunkified to a smaller peptide, and then that peptide undergoes a couple modifications that modify it a little bit. So that is what GLP-1 structurally is. Okay, so in the 1980s, they discovered the gene that encodes for these hormones. And what it does is it's secreted in response to ingestion of nutrients in the gut. So when you eat something and you collect nutrients in your gut, that will cause the secretion and the production of GLP-1. So it's associated with, or let me ask it as a question. If you eat a bunch of food, what does your body then want you to feel? Yeah, you want to feel full. So your body is going to make hormones that then respond to the stimulus of you eating the food so that you feel full, so that you don't constantly keep eating. Okay. So that is essentially what GLP-1 does. It helps you feel good. It helps you feel full. So obviously, if there is a GLP-1 peptide hormone, what do all hormones have that they bind to? A receptor. So there's a GLP-1R, which is the GLP-1 receptor. Okay, and this is a membrane protein. So most receptors are membrane proteins. And so again, if you have a cell, nucleus, whatever kind of cell this is, oftentimes the cells will put protein receptors in their membranes that can sense the extracellular environment. Okay, so there is a GLP-1 receptor, which is part of what's called a G protein complex. I'm sure I get the word right. G protein coupled receptor. You hear about these a lot in biochemistry. Who took biochemistry? Did you hear about G protein coupled receptors? Cell biology. Okay, so you'll learn more about that in cell biology. Essentially, there are these large family of receptors that all of them are designed to bind to some chemical X that's in the extracellular matrix, and when it binds to the G protein coupled receptor, that initiates some signaling cascade. So it activates the receptor, which then causes some enzyme to activate, which then activates some other enzyme, which then activates some other enzyme, etc. Oftentimes, you'll see a chemical cyclic AMP increase if a G protein coupled receptor is activated. Okay, so the receptor for GLP-1 receives a signal and it turns on some chemical cascade inside of the cells. And this acts to regulate blood glucose levels and lipid metabolism. And here's an interesting tidbit. So if you create a GLP-1 receptor knockout, so you knock out the receptor, that leads to increased appetite, contributing to weight gain or obesity. So you can actually get a mutation that causes you to eat too much and you gain too much weight because you never feel full, essentially. Or I don't want to say you, but the creature that has this knockout, they never feel full because the GLP-1 cannot interact with the cell because the receptor's gone and it cannot activate the signaling cascade. So these hormones have to deal with how you feel full, and the receptor turns on the signaling in the cells to make you feel satiated. Okay. So then the next term you need to understand is a GLP-1RA. So this is a GLP-1 receptor agonist. So agonist is a biochemical term. If you take biochemistry or if you take maybe pharmacology, you'll start to learn about agonists. So an agonist is going to be an analog chemical that is not the exact same chemical, but it binds the receptor and causes the same action. So it's essentially a mimic of the chemical X. That's an agonist. So a GLP-1RA then, receptor agonist, is a GLP-1 mimic. Okay. And people are interested in these mimics as drugs because they can delay gastric emptying. So if you have food in your stomach, they can cause it to sit in your stomach for longer, making you feel full for longer. They can reduce the food intake, so they're behaviorally modifying your behavior, make you feel full. And they suppress your appetite, these receptor agonists. Okay. And then the final enzyme that you need to know about to understand the whole process is this called DPP4, dipeptidyl peptidase. So this is an enzyme that degrades GLP-1 in the bloodstream. So imagine you eat a meal, you take in a bunch of nutrients into your gut, your body produces GLP-1 peptide hormone, and then you feel full. Why does the GLP-1 need to be degraded? Yeah, you're going to get hungry again. So you understand that the whole body is always cycling through these homeostatic processes. If the GLP-1 just floated around in your blood forever, you would feel full, you'd never eat again, in theory. So you need an enzyme that degrades the GLP-1 so that you then start to feel hungry again at about the right time. So why is this kind of a problem for the medicine? So if you were the simplest way that you could maybe create a medicine around this is, why don't you just inject GLP-1 into people? Like, why wouldn't that just work? Knowing all these terms now, like, why do we have to create these receptor agonists? Why don't we just inject GLP-1 in obese people? Yeah, your body's just it has this enzyme and it's just going to degrade the GLP-1. So they originally did try to use GLP-1 as an enzyme. The problem with GLP-1 is it's got a very short half-life. So it's degraded very, very fast because physiologically you want that response to go away. You want it to be temporary. So it makes sense that then the actual quest of what needs to be done is somebody needs to find a GLP receptor agonist that cannot be degraded by dipeptidyl peptidase. That would cause you to feel full for a very, very long time. It would suppress your appetite for a very long time. Okay. Um, I have here, so why would you want to do this? Being fat is a problem. Let's be honest, we don't want to be mean, but it causes cardiovascular diseases, diabetes. It's not very healthy. So if we can, I mean, the best way in theory would be to have people exercise, but if that's not working, then maybe we can develop some drug that suppresses people's appetites. That was I feel kind of cynical about like teaching this. Like it makes me cynical about life that people cannot just exercise and diet and get healthy, that we need to sort of like create a drug that does this. So I'm not sure how I feel about that, but it's happening and it was very successful. Okay. Let's see. We already talked about this. We already talked about this. Okay. So the quest was to find a GLP-1 receptor agonist that is long lasting. So then comes in sort of like the biotech side of this, where I don't want to say it wasn't random. They were actually looking for enzymes that could activate these receptors, but they were looking in Gila monster spit. So here's a Gila monster. I think they have these in Arizona. I think I've probably seen a few of these when I was a kid in Arizona. The species is Heloderma horridum. Okay. And when you read these papers, there's a couple of them. Let's see. I pulled out some pertinent quotes. Let me just look through these. An amino-terminal histidyl structure is characteristic of most peptides in the glucagon family. That's hard to understand, or it's just a lot. All that means is that what's characteristic of the glucagon superfamily is that these peptides start with histidines. So if you look at this peptide, it starts with a histidine. So how is that different from most proteins? Yeah, most proteins are going to start with a methionine. Okay. And how does the histidine actually get there in the beginning? Because ATG is the start codon. So how can you even get a protein that doesn't start with a methionine? Yeah, because it's derived from a chunk. So this is another theme that you'll see in physiology of peptide hormones is that if you need something that starts with something other than a methionine, usually it comes from a gene that starts with a methionine but gets cleaved as a precursor into the active form. Okay, so because these are kind of a unique family of peptide hormones, they're small and they start with histidine. People started developing experiments to try to look for small peptides that start with histidine. And there are some sequencing methodologies for proteins that we'll talk about in a second. So the next thing I pulled out here, it says an amino acid sequencing assay for peptides containing an amino terminal histidine residue was used to isolate a 39 amino acid peptide extended. So let me just talk. I'm not going to talk a ton about this, but I'll explain just enough for you to understand what's going on. So there is technology for sequencing proteins. Normally, when you think about sequencing, you think about DNA, A's, T's, G's, C's. I've covered a lot of how they sequence DNA. Sequencing DNA is obviously going to be a very different biochemical process than sequencing proteins because it's a totally different chemical grouping. So, but there are technologies to sequence proteins. So one of these is called Edman degradation. And again, I'm not going to explain all of this, but I'll explain the basic concepts. So if you have some protein that is a linkage of amino acids, in Edman degradation, what they do is they can put a modification onto a terminal group, and then they can induce with an enzyme a cleavage event that cleaves off this last piece. Okay, so then they will produce this peptide and this amino acid with some modification. And there are mass spectrometry techniques that allow you to measure the mass of this amino acid that gets cleaved off. And every amino acid is going to have a unique mass because all the amino acids have different R groups. So you can actually determine what amino acid is there based on essentially its mass. And then, so Edman degradation works by iteratively doing this. Excuse me. They're iteratively cleaving the final peptides with iterative reactions, and then they're getting this peptide sequence. And so what they're doing is they're extracting saliva from the Gila monster, and then they're just doing Edman degradation. And then the things that they're interested in are the ones that start with a histidine. That's essentially what they're doing. And so then they're getting sequences of abundant peptides that are in the saliva that start with histidine. That's all they were doing. Yeah, they measure it by mass. In more complicated, what's called tandem. So okay, let me actually just go into this. It's probably worth talking about this tangent a little bit. In tandem, in liquid chromatography, tandem mass spectrometry. People don't really do Edman degradation anymore. That was kind of like an old thing. Most sequencing nowadays of proteins is pretty much done with LC-MS-MS. So the abbreviation for this is LC-MS. MS stands for liquid chromatography tandem mass spectrometry. Essentially, what's happening here is something very similar, where if you have a protein, which is some number of amino acid linkages, typically what they'll do is they will add enzymes protease enzymes that will chop this enzyme up into little chunks. So if you add, for example, trypsin, does anybody remember where trypsin cleaves? K's and R's, lysines and arginines. So if you added trypsin to some protein that you want to sequence, it's going to cleave it, and it's going to make all chunks that begin and end with either a K or an R. And it's going to produce these various chunks, okay, these peptides. And then what they can do with these peptides is they can ionize them and break them. So they break these smaller peptides, again, breaking them into little chunks. And then they can ionize these so that they become gaseous in a mass spectrometer. And then they can measure a mass to charge ratio. And they can actually measure like time of flight. So they measure how fast they travel in the spectrophotometer. And from that, they can derive a mass to charge ratio. If you wanted to actually understand how they're actually doing this, this would be a graduate level biochemistry class or a graduate level chemistry class. But just understand that there are methodologies to sequence proteins. The modern age one is LC-MS-MS. The older one is Edman degradation. And in this case, they're using an Edman degradation style analysis. Sure. Yeah. So I mean, so these are the sequence. Here's the sequences. Let's talk about that. So this is the sequence that they got. These are the two sequences from the Gila monsters. They call them extended. They did not have a genome for Gila monster. I don't know if they have a genome for Gila monster now, but if they did, and you have this sequence, how would you then find the gene for that in the Gila monster? What would you do?
I guess it's multiple combinations.
Well, there's a blast algorithm that does this. So there's only a certain amount of codons that can code for each amino acid. So you can do what's called a T-BLAST-N. T-BLAST-N is on the BLAST server, NCBI server, where if you want to blast a protein against and find its gene, you just take the protein sequence, you do a T-BLAST-N. That will find its amino or its gene sequence. And you can find the gene sequence by just essentially translating backwards to the possible codons. You don't know exactly what it's going to be because there's going to be various codon options, but then once you find the gene, you'll figure it out. As long as you have the genome, it's easy. Okay, so here is a list of the proteins that they produced. These extendins, these are the glucagon agonists from the Gila monster. And they compare it to glucagon. So here's the human glucagon sequence. And here's a question. Is this an ortholog or an analog? So if you look at what's conserved, the boxes are what are conserved here. So there's a conserved histidine in the beginning. Then there's a conserved GTF, TSD, this thing. And then these are conserved, and then this, and this. So there's only about, in the paper, they say it's about 53% conservation. So what do you think that this is an ortholog or an analog? And do you know the difference between those? Okay, so then let's review those terms. So remember, for proteins, there are homologs. Within homologs, with a subset of homologs, there are orthologs and paralogs. What's an ortholog? Who remembers? Other species, same gene. So other species, same gene. So chimp hemoglobin versus human hemoglobin. Paralogs are same gene duplicated in same genome. So paralogs can evolve a lot of diversity, but if they're evolving, you're probably not going to see 53% diversity of a paralog, where essentially 53% of the amino acids are not the same. In an ortholog, you're also probably not going to see, 50% homology. It's essentially saying that this alignment is 50% homology. That's a very, very low homology. There's probably just some protein domain that is conserved, but the main point is that these are not conserved enough to be considered orthologs, so they're not descended from the same gene family. So it's not as if the Gila monster is working with the same gene in its spit as humans are in the GLP-1 gene. Does that make sense? Like those are not the same gene, but the same evolutionary history. What's happened is that just through convergent evolution, they've derived a similar sequence. That's what's called an analog. So an analog is not technically a homolog. It's just something that has arrived at a similar structure based on convergent evolution. Okay, so in this case, it's a small peptide. It's probably honestly just by stochastic randomness. So the main point is that the Gila monster spit is not the same protein as GLP-1. It's just an analog. It looks similar. Okay, so then once they had found this theoretical GLP-1 analog, what would you want to do with it? These Gila monster ones. What should you do with them if you're trying to do drug development? Yeah, you want to do a test to see if it does the same thing as the glucagon GLP-1 peptide. So they did some of these tests. Okay, this is a real old paper. It's kind of like old school techniques. And here they're measuring cyclic AMP. So cyclic AMP is a measurement of that G protein coupled receptor activation. And these are on pancreatic cells. So actually what these are called, is kind of an interesting experiment. In the methods, it's called dispersed pancreatic acini. I don't know how to pronounce that. But what they did is they collected cells from guinea pig pancreases. So they essentially would cut out guinea pig pancreases. They would digest around the pancreas to collect the pertinent parts of the pancreatic cells that they wanted to analyze. And then they would take those cells and then they would treat them with the essentially the Gila monster peptides. Okay. And what does the data show when they treat the pancreatic cells with these Gila monster peptides? What's happening to the cyclic AMP levels? They're increased. It's not hard to interpret. They increase, they go up. So essentially they have a little bit of mechanistic data showing you that these things are active. If you put them on cells, you'll get an effect that's similar to GLP-1. So this is how they first came to the idea that these might be GLP-1 agonists. They might be activating the same receptors, same type of activity. Okay, let's see. So that was all. What year is this? Did I get the year? That was 1990. So this is in the early 90s that they're doing this Gila monster stuff. Then just recently, I don't want to say just recently, but maybe like 10 years ago, they started developing semaglutide, which is a new derivative of these GLP-1 receptor agonists. So essentially what the breakthrough with the Gila monster stuff is that these GLP-1 agonists were longer lasting than the GLP-1. So the big breakthrough with the Gila monster thing was essentially like we can discover other agonists that do the same activity, but are not subject to the same degradation. And so they should be able to last longer. And so that kind of let people realize, well, there might be other things out there that we could discover that do the same effect, but might be even longer lasting than the Gila monster ones. Okay. So then people started exploring all kinds of different chemistries for GLP-1 agonists. And the one that's in Ozempic, so the Ozempic is like the newest drug that everybody's heard about, or maybe you haven't heard about, but that's now big in the news that people are taking that's pretty successful. The chemical inside of Ozempic is called semaglutide, which is a GLP-1 receptor agonist. Okay. And this is what it looks like. So this is from the patent. This is what the semaglutide peptide looks like. What do you notice about it right away? It starts with a histidine. It's got it's got this TFT thing. It's got this conserved domain. What else do you notice about this peptide? Yeah, it's got some real weird stuff. So here's something that's very strange. It's got this. Is this in our group? What is this? What? So this is essentially a different chemical moiety attached to the lysine R group. So the lysine has an R group. Okay, and then attached to that lysine R group is this moiety, whatever this thing is. And I think it says what it is. It says the 26th lysine of the peptide sequence is connected with a C18 fatty diacid. So this is a C18 fatty diacid. So first thing very strange to notice about the structure of semaglutide is that one of these lysine residues on its R group has attached this fatty acid thing. Okay, what's the other thing that's very strange about this peptide? There's one other part of this that's you should not have seen before, and you guys have drawn so many amino acids that what? No, I mean you would expect to have a COOH at the end of protein. That's not bothersome. Where's the other thing that you see something that's very strange that you should not expect to see in a protein? Where?
Second.
First say left or right? On this? On this? This. This is definitely something very weird. Okay. So this is actually a non-canonical amino acid. So I'm glad that you found it. So this is the other weird thing about this. In the text of the patent, it says this is a his-abe, and it says, so this is a histidine connected to an AIB. AIB is an unnatural amino acid. Okay, so I looked that thing up. This is what it is right here. ABE is alpha amino isobutyric acid. So in this class, I made you memorize the 20 canonical amino acids, the 20 amino acids that life uses. There's actually other amino acids that life does not classically use. And this is one strange non-canonical amino acid. Okay. So now let me ask a question in terms of like protein engineering. Why might it be helpful to put a non-canonical amino acid in a protein? Yeah, so it's going to be the actual amino acid sequence that causes it to be recognized by DPP4, which is going to cause it to be degraded. So if you start changing the chemical in a way where you're changing it, making it resistant to be bound by it, the thing that cuts it up, DPP4, it's going to last longer. So what they say in the patent is that this non-canonical amino acid is actually what's causing the resistance against cleavage. This is actually the site that is causing it to be resistant to be cleaved against DPP4. And it has something to do with the fact that this is non-canonical and these two carbons, they cause steric hindrance and they resist mobility in that particular spot. And for whatever reason, that causes it not to be allowed to be cleaved by DPP4. Yeah. That's a great question. And that is the next thing tried to research this. So how do you produce this? That's literally the next question. Very good question. So if it's non-canonical, one question is, okay, so can we produce this with recombinant protein expression? What? Well, it went through FDA screening and it was not toxic. So, I mean, you would wonder, like, well, is it? I mean, it's definitely like unnatural. I don't know, but like, so again, like, would I take Ozempic? No, and I think that there are, I think that there are. I have to be careful what I say. I don't want to say anything negative about. I guess I don't want to be legally ramified for something I say about a drug, but I'm sure that there's side effects to all drugs, right? And so there might be some side effects from eating a chemical that has non-canonical amino acids. Yeah, for sure. Yeah, exactly. Yep. I don't know. You'd have to look into that. But essentially, okay, so all drugs are going to have side effects. So is there going to be some negative connotation to eating this drug? Yeah, for sure, probably. But the question is, does the side effect outweigh the benefits of, if you're 400 pounds overweight and it's like, is this going to kill you or are you going to die because you're 400 pounds overweight? Like maybe then you'd rather take the drug. Like that's the calculus in all drug development, not just this one, but in all drugs. That's the calculus. Is it all drugs are bad on some level, just like you can die from drinking too much water. Okay, so the next question is, how do you mass produce this? So my question was, can you produce this strange chemical with recombinant protein expression? Yeah, you won't be able to produce the final form of this with recombinant protein expression. What part of this could you make with recombinant protein expression? What part can you make? So if like I was to highlight the part that you can make, what part can I make? You can make this part. This part is just standard canonical amino acids. So this is essentially going to be a 3-step synthesis. So the 3 steps are going to be 1, recombinant protein expression of this blue thing. And this is what the patent talks about. So it's going to be recombinant protein expression of this blue thing. And then I don't know actually what order, 2 or 3, but then there's going to be a process by which you chemically, so you're using chemistry reactions, like organic chemistry reactions, to add on the his AIB onto the N terminus. And then you're going to have to add this C18 fatty diacid onto the lysine. So it's essentially going to be a 3-step synthesis. And actually there's so again, I did a little bit of research on different ways to produce this. There are also other companies that just straight up produce this with straight chemistry. So not all, because it's a small peptide, they can develop chemistry reactions to just sort of synthesize it. And so right now, I think they're trying to figure out, is it more cost effective to just produce it with chemistry, or is it like pharmaceutical chemistries, or is it more cost effective to produce it with recombinant protein expression? That's kind of what some of the recent research is. Yeah. You can't, so you can actually genetically engineer microbes to introduce non-canonical amino acids. So there are genetic systems where you can give bacteria certain tRNAs that will allow them to couple non-canonical amino acids. So that actually is possible. And there are people who study that. There are people who develop like non-canonical DNA codes, and that is possible. Although it doesn't seem like that's possible for this particular one. But that is possible. And there are people that do that. Okay, so then I guess just to finish up, I looked at the plasmid. I looked at the plasmid that's expressing this, just to learn a little bit about the plasmid. So where do you think that they're expressing this peptide in? Based off this plasmid. Is that what you think? What makes you think that? It's got a bacterial origin. It's also got an AMP cassette, but what else does it have? Well, it's got restriction enzymes. Yeah, all plasmids are going to have restriction enzymes. But what makes you think it's E. coli? What else could it be? Look at the other features and remember back to what I taught. What else do you see? There's a 2-micron origin, which is pertinent for what organism? Yeast. So this is a yeast plasmid. And what else do you see? If you ever wondered that whether it was yeast or not, what else do you see in the sequence or in the features that tell you immediately, okay, this is a yeast plasmid? S. pombe. Anybody know what S. pombe is? That's the fission yeast. So this is a species of yeast. So we talk mostly about Saccharomyces cerevisiae, but S. pombe is a fission yeast. So they're expressing this in fission yeast. And fission yeast is, again, the reason you might do this is because they probably want some kind of modifications that are capable in the yeast, some post-translational modifications. Or maybe they're doing it because they like it to be expressed and secreted. That might be one reason to do it in pombe. Okay, but here is the plasmid. Essentially, you should be able to look at this plasmid and kind of understand it. Like, here's a promoter. Here is the GLP-1 peptide that they're expressing. I'm assuming that's this. There's a terminator. There's some restriction sites. There's pombe promoter stuff. There's the 2-micron origin. That was the key to understand that it was yeast. And then there's E. coli stuff, like any other standard background. Okay, so that's the plasmid that they're using to overexpress this in the patent. Okay, final thoughts. We won't go too hard on this, but final thoughts is one thing to point out that was interesting in my research on this topic is that this started in the 80s. And so this is a 50-year research trajectory to get to the current modern drugs. So people don't understand how long research takes. You know, it was somebody's 5 to 10 year life just working with Gila monsters, like figuring out what proteins were in the Gila monster spit. And they probably had some idea that like they were looking into these GLP-1 agonists, but they probably did not really know that many years later it would lead to, you know, a great drug discovery. Maybe they had some inclinations, but it takes a very, very long time. That gets, yeah. So the question was, why would somebody even be looking at the Gila monsters? And that gets to this question, which was, I was going to kind of talk about, like, is this basic or applied research? Basic research would be like you're just interested in something and you're looking at it because you're interested. Like, it might be worthwhile to know what kind of venoms Gila monsters have. And studying venoms is actually like there's many people who study venoms, like snake venoms, bee venoms, insect venoms, and actually venoms are a source for many drugs. So you can find in snake venoms, there's lots of blood thinners that people actually have used that have been FDA approved to thin blood. So there's lots of medicinal purposes from venoms. So that might be one reason to just, from the basic science, to just study venoms. Like, we want to know what's in venoms. The other side of this is that I did look into that question, like, why were they studying the Gila monsters? And their listed rationale is that there might be something interesting about Gila monster like guts and saliva, because they apparently can eat a ton of food in like one sitting, and then they don't eat for a very long time. So it seems like they probably had a hypothesis that maybe there's something special about the glucose signaling in the Gila monster. And so they looked at it and then they kind of got lucky and found this peptide. And so that would suggest that it was kind of like applied. Like they did have a research tech that they were looking to try to get into. But that's always a conundrum of like basic versus applied research. It's valuable to do research on things because you're interested, because you never know 50 years from now, somebody might utilize your discovery and turn it into something great. You never know what's going to happen. And research takes so long that even in somebody's lifetime, you don't really know where your work's going to end up. Okay, so that's the end on GLP-1 biotech and drug development. That's the best I could do for drug development lecture. Hopefully that was helpful. Okay. Yes. Okay, have a good day.